in binding to BEL5, how it is moving. So what we did, we removed those two UTRs from the BEL5 and again generated plants and tried to look at its mobility. And we do see there is a very less mobility, absolutely less mobility of the BEL5 RNA, suggesting that those two UTRs probably played critical role. If that is the case, now I kept the two UTRs intact, put a junk RNA inside, removing the BEL5 from the construct. Okay, this is the beauty of the experiments. You keep both the UTRs intact and put a junk RNA inside. We do see junk RNA is moved, suggesting that my two UTRs are indeed playing major role. We looked at the UTRs. We do see there is loaded with polypyrimidine tracts. You have polyUs located in the UTRs. Okay, so these are the old experiments we are just sharing. With the help of those Q-carbide protein that was identified in 2009, we looked at the potato genome. We identified 6 PTBs, polypyrimidine tract binding proteins. Polypyrimidine tract, RNA has polypyrimidine tracts, and these proteins bind to this RNA. So we identified 6 of them, and these 2 look like the one that Q-carbide, the UC Davis group identified, and we have again worked on these 2 proteins, PTB1 and PTB6. Now, if you take these proteins and express to the plants, what you like to see? What is the observations? Okay, by the way, this is the human this is the human PTB1, and we have looked at that it has RRM1, RRM2, RRM3, and RRM4, the RNA recognition binding motifs where RNA binds to proteins. So what is the function of these PTBs? Why do they bind to RNA? And I said this is the prediction. Yes, we did the RNA EMSA. We showed that it binds to proteins. Where are these proteins expressed? And do they bind mobile RNAs? These are many questions that popped up during the studies. Now, if you look at, I keep saying the polypyrimidine tracts. If you look at a particular 3' UTR of PTBs, you can see this is the BEL5, I think. Yeah. So you have a lot of polyUs in between. Okay, and this is where the proteins are likely to bind. We did RNA EMSA, and in RNA EMSA it was shown that the PTB1 binds to BEL5. So suggesting that PTB1 probably they're escorting the BEL5 from leaf to the stolon transport. Now, if we express any of these PTBs, I have a few minutes, 5 minutes? Okay, I'll be done. So, if you express PTBs in the plant, if you overexpress PTBs, and now we look at what happens. If you overexpress PTBs, it increases tuber yield. A protein which is a transport protein, which is transporting an RNA molecule, you are expressing in the plant is making more tubers, number 1. Okay, it's also expressing the BEL5. So what is the suggestion is that this protein is basically stabilizing the BEL5 function. Okay, it's making more tubers. It's also like a BEL5, it's an activator. Okay, so we looked at all these. We also looked at the target RNA. We looked at these RNA expressions in the plants and see the conclusions what it is. So STPTB proteins enhance both STBEL5 activity and movement in potato plants. You have another protein, PTB6. If you look at PTB6, I think it's the RNA suppression line. If you suppress it, you see completely no tuber, suggesting that this whatever you got in the overexpression phenotype, in the knockdown phenotype, you have exactly the reverse. So it's suggesting that it is also a positive regulator for potato tuberations. We looked at its expression. It's beautifully expressed you can look at it, phloem parenchyma. It is expressing in there. You look at how beautiful this protein is, STPTB1, how its expression, suggesting this protein is binding to BEL5. It's expressing in the right cell and taking the RNA to the target organ. The potato genome got sequenced 2011. We looked at more bells. There are more bells in the protein. Now we have, I think, 14 bells in the protein genome, and we wanted to look at 2 more bells, which are very close associations with respect to its amino acid sequence with 11 and 29. I can see it here, 11 and 29. These are the 2 RNA molecules. I already explained to you the STBEL5 is a positive regulator. Now I took STBEL 11 and 29 to look at further because of its close association with BEL5. We thought it will have redundant functions. Now when you overexpress this, the expression pattern of BEL 11 and 29 does it affect? We looked at its expression, by the way. So what does this RNA express? And if you see this is the stolon. I can see from here. This is the short day and this is the long day. Short day is the inductive condition. So this RNA get highly accumulated, suggesting that they may be acting like a BEL5. Okay, so we overexpressed. Okay, you can skip these results. These are also expressing in the phloem parenchyma and companion cells, and we overexpress this BEL 11 and 29, the two other transcription factors, their RNA molecule, and we do see they have reduced tuber yield. Unlike the BEL5, they have reduced. That means they are repressing the tuber activity, right? Okay, good. If you suppress it Do they move? Okay, yes, we also looked at, do this RNA molecule move? And we proved by the same grafting technique, they move by a different promoter technologies. We proved that these RNA molecules are also indeed mobile, 11 and 29. Now, interesting thing is, if you suppress this, where is that gone? Yeah, if you suppress this 11 and 29, they make twice the number of tubers. So these two are a repressor, whereas BEL5 is an activator. Now, BEL5, 11, 29, they have pretty close amino acid sequence similarity. You would expect them to have the similar functions, but one is activating the process, other two are repressing the process. Why would you guess it? Why would it happen? Okay, so this is, we looked at its, as I said, these two are also mobile. BEL5 was also mobile, 11 and 29 was also mobile. Now, we looked at its RNA molecule, we looked at its UTRs. They do have the polypeptide tracks. We do not know which protein binds to this. We are now working on this. But at least in the case of BEL5, we know PTB1 binds to it. Now look at this diagram. This is that cartoon and where I'd like you to focus and take your attention here. So this is, as I said, this is nothing but a shoot apical meristem. And this shoot apical meristem, if it senses light, it makes the plant. If it doesn't sense the light, it makes the potatoes. So what is happening? In this shoot apical meristem, we are giving the analogy of that you have three RNA molecules. One is a activator and two are a repressor. 11 and 29 are repressor. And why would you think it will work in this way? Please remember this, before the onset of tuberations, this organ, what happens before the onset of tuberation, it stops to elongate further. Plant is likely to kickstart the tuberation mechanism. It stops the elongation. As a result, what happens? There is between the pith and cortex, the growth initiates, and this growth expands. As a result, you get to see a swelling of this organ, and this swelling expands, and then it accumulates the starch. As such, the swelling starts. You can imagine there is a tremendous change with respect to its radial versus longitudinal cell division pattern. Earlier, it used to be a radial expansion, and it was not. It was a longitudinal, now it will be all radial cell division pattern. So with respect to this 11, 29 and 5, we now explain this fact, these three transcription factors or their mRNAs are expressing in these cell types and governing these complete cell division patterns, and thereby making these tolerance to, not only it has to target numerous target genes with respect to hormones and many things, right? So you can expect what is going on here. Right? And this activator repression mechanism is not new, not that we are first time saying. If you look at flowering with respect to shoot apical meristem, if this is what I said, exactly what is happening here, right? If you look at a shoot apical meristem, and shoot apical meristems, I'm sure many of you may be aware that how shoot apical meristem, the stem cell is governed in the plant cell, how the stem cell number is governed, how does plant accomplish indeterminate growth with respect to maintaining the stem cell between the activation repression mechanism, inhibition mechanism between the ocell and clavata. Okay, so that mechanism is known. So we are trying to understand what is happening with respect to this stolon organ. This is what it is. I can skip. So is the flowering. If you take FT, just one more minute. If you take FT is a flowering signal. If you make one amino acid change in FT, it becomes a repressor. Usually FT is an activator of flowering. One amino acid you change, it becomes a repressor. TFL is a terminal flower homolog. This is an inhibitor of flowering. Okay, so this inhibition mechanism in terms of shoot apical meristems, in terms of flower, in terms of potato this stolon development. I said this stolon from stolon to the complete potato development is a complete dynamic process. We are yet to understand that how does it work. Okay, so in summary, what I say, BEL5, 11, 29 could function collectively as phloem mobile signals in a whole plant network in potato that modulates storage organ development through the process of cell growth activation and suppression in the subapical portions of the stolon tip. Okay, so there is a lot going on there. I can skip this. This is the total summary that we have recently written on review in Plant Phys, that how does a potato tuber made. You have a lot of activators are expressed in the leaf. These activators, I did not explain the measure. Another one recently identified 2011, SP6. SP6 is a co-regulator. Okay, it works along with BEL5, and these all signals, it works as a protein. It goes as a protein signal. This one goes as an RNA signal. They all require to go here and turn on the battery of the tuberations mechanism, thereby the tuberations, and work with numerous target genes in this organ. So for me, this is the reaction center. There is a lot going on underground, below ground that we do not know. We are yet to understand. Okay. So my lab is, I collaborate with many people in India as well as in abroad, and these are my generous funding from all of these funding bodies are gratefully and acknowledged, and I thank all of them for their support. These are my papers in last 9 years, or even some of them from postdocs. This is my group in ICER Pune. You are most welcome to visit our institute. And if my concept is whatever I discussed, I like to ask these questions when all experts are sitting here. If the concept of mobile RNA is indeed true, and if it is working that way, these all organs that you see, we are eating all modified, is either modified root or modified stem, or you consider all this now. This was the root before, it became a modified root. Consider this, it's a modified root either. So what's going on here? I'm pretty sure in all these cases, a sweet potato we don't work much, but you can imagine with respect to its energy demand, it's so important crop. So we are paying little attention to it, and we're trying to understand, does mobile RNA really work in all these storage crops? Okay, thank you so much. If there's any questions, I will be happy to answer.
Any questions?
Thank you for the nice presentation. Sir, what are the genes responsible for BEL5?
I'm sorry?
Sir, which genes are responsible for the production of BEL5 protein?
What genes are
responsible for the BEL5 production?
BEL5 production?
Yes.
Acha. So upstream of the BEL5 we have not worked out. It's BEL5 we identified from a cDNA library. Long back we made a stolon specific library. From library we did a yeast 2 hybrid skin, so we identified BEL5. From the Arabidopsis BEL, we took Arabidopsis BEL as a query, and thereby you pulled out the BEL. These are all transcription factors. They govern many other genes. That's what we do.
Hello, sir. So
yes.
This BEL5 activator and repressor RNA.
BEL5 is an activator. Repressors are the 11 and 29.
Yeah. Will the three travel together?
Very right questions. I said in summary that it makes sense for me because the subapical stolon is to develop, and I said there is a radial versus longitudinal cell division, complete cell fate change, and these three may be expressing there at a different cell types and governing the different genes with respect to new cell division pattern. Imagine the stolon, which was earlier, it stops elongating, right? It doesn't elongate anymore. It stops just before the onset of tuberization. It stops elongation. Now it swells up. This is the situation, right? It's swelling up, and this is what we think all three RNA are being mobile. Not only all these three. I mean, there are many other pathways are required.
What I'm asking, all three is moving together or what? That's the question. Because if they are moving together, then the same protein must be taking that. Because you didn't find the protein which is taking that.
See, I showed you this plant requires an inductive condition. Under inductive conditions, if you go and test, you will find all three. You will find all three because they are required to go. In the juvenile stage, we don't get to see this, by the way. We see when the plant is becoming the mature and about to start the tuberization. So we see these RNAs are. And these RNAs, they don't, we have looked at their stability as well, and they're highly stable in the leaves. That makes sense that they are the mobile RNAs.
Good evening, sir.
Yes.
Sir, I just have a curiosity that you have worked upon the different activators and repressor, how they regulate tuber formation.
Yeah.
So is it possible that by playing around the genomes using different genome editing tools, etc., we can make tubers above soil? They are normally formed below soil. So is it possible? Because nowadays we are playing around genomes.
I can tell you nothing is impossible. One thing, as you have seen all the great talks before me. Who am I to? But I told you clearly that by simply manipulating one smaller array, 56 microRNA 56, right? We showed the plant develops all the stems not below ground, above the ground, and they are all making tubers. I'm sorry. The test is yet to be tested. Professor Krona will not let me go like this. He wants an answer. So we are yet to test the nutritional quality. Yes.
Do you know what RNAs are doing? How do they work?
Do I know these RNAs are?
How do they work?
What are they doing? So in the stolon, what they are doing right now, we do not know. But we have looked at their common targets. We have looked at their all the marker genes that they target, and they all. That means they are there, they are going there, and they are activating those target genes, numerous target genes essentially involved in the hormones. Most importantly, gibberellin. GA is a classical hormone. GA must go down before plant starts the tuberations. And we see the response genes that they target. And they target these RNAs, they make the proteins there. And then they, in tandem, they bind to the target genes. There is it's called, they make all these autoregulations of their target genes, GA20 oxidase target genes. They bind and regulate the GA level there. So they get translated there, not in the leaf. They all go there. That's the question.
You not say that RNA is translated.
RNA is translated at the stolon, below ground, not in the leaf. Not in the leaf. And even if you see these RNAs are translated at a leaf, it's a very, very negligible level. We see probably tenfold more proteins. Of course, we are yet to see with respect to the antibodies, but we have seen at polysomal level what is happening of those RNAs.
Okay, we have one last question.
So, the movement of RNA is through the RNA binding proteins.
Yes.
So when they go to the target, they have the RNA is need to be translated. So how the dissociation of those protein is occurring?
So I encourage you to look at the plant cell paper 2009 from UC Davis, the first paper in the field. No, I'll tell you the answer. Answer is this: all these proteins, they do not remain anchored with the RNA anymore. As long as the transport is there, they remain anchored. We have seen the reports clearly describe that at different point of analysis they have seen not all 16 remain anchored. They have got shed off at the transport, but at the target organ 1 or 2 proteins finally still remain. That's what they have shown beautifully.
Okay.
So they get
Thank you.
Okay, we have one more student question.
It's good. Let students ask.
Sir, is there any signal from shoot apical meristem?
There is a signal from is there a signal from shoot apical meristem?
To the RNA, to the
Stolon?
proteins to go to take RNA to the stool.
So these PTB proteins, which are polypeptidine tract binding proteins, I did not show their expression pattern. They express. At the same time, when RNA is expressed, if you look at the PTB proteins and their expression patterns, it makes sense for you to understand that they are also in the shoot apical meristems and stems, and they are expressing, right? They are expressing in the phloem tissue, so that they can pick up the RNA molecule and make the complex and then do. We did many molecular assays that they indeed bind and they express.
Because potato tubers are accumulating starch.
Yes.
So usually after flowering, starch gets accumulated in the grains. So I was just thinking in that way.
No, so plants, before they go for flowering, it will induce the tuberation. It will induce the induction of protein.
Like cereal grains,
Uh-huh.
in that case, if the flowering happens,
So flowering.
starch accumulates.
So flowering in cereal clays, as Professor Khurana was describing, in rice, you can see beautiful signal. Now we know which protein goes and signals rice to flower, right?
Okay, I think we'll have to stop there. Please join me in thanking Dr. Banerjee for his presentation.
Yes, just get up here with the mic. The final talk of this symposium is by Professor Michele Lovan. Yeah, you want this?
Yeah, I'd prefer that.
Oh, yeah.
Okay, we made it. To everybody. Okay, and we'll see if I can do this a little quicker than planned. So is that working there? So I'm going to tell you about a project that I only just really started recently. It's very much a work in progress. And it's regarding a role for the Fusarium graminearum ST2P receptor in mediating the wheat pathogen interaction. So Fusarium graminearum is a necrotrophic fungi, and it is the causative agent of Fusarium head blight in cereals. Whoops, I see the button problem. So that would be, okay, so spores of Fusarium graminearum, they float around in the air. They will land on a healthy wheat head and immediately or shortly thereafter germinate And the hyphae will grow and infect the rachis, which is kind of like the internal plumbing system on the inside of the wheat head that brings all the nutrients and water up to the head for the developing grains. When the rachis is infected, it turns off that flow of nutrients and water, and the kernels start turning yellow and stop developing and eventually shrivel up and die. And this leads to dramatic decreases in yields for the farmers. These are just some fusarium spores. So the other problem with fusarium is that, or graminearum, is that it also deposits mycotoxins. In particular, it deposits deoxynivalenol, which is known as DON or vomitoxin. And while it's part of the infection process that allows this to be a necrotrophic fungi, it's also bad for animals and humans who consume the DON-contaminated grain. It leads to horrendous vomiting, and people have died of dehydration and whatnot from it. So it is a problem. Historically, we've not had fusarium head blight in Canada, but because our main wheat growing area is in the western provinces, Manitoba, Saskatchewan, Alberta, where it's quite cool and relatively dry. But with climate change, we're seeing more humidity and warmer temperatures generally, and with that we're seeing a significant increase in the occurrence of fusarium head blight in Canada, most notably over the last 10 years. So to address this and a variety of other issues with wheat in Canada, the Canadian government in 2012 initiated what we call the Canadian Wheat Alliance. And it brought 53 principal investigators together from 4 or 5 different institutions with the mandate to improve the yield, sustainability, and profitability of wheat. And they asked if I would participate in that, and I said, sure, maybe I can help with fusarium. But there's other members looking at all other aspects of improved wheat in Canada. Okay, so on to the molecular. So this project that I just initiated recently in this context is related to the ST2P receptor of the graminearum pathogen, which is a G protein coupled receptor. It is ST2P and its close homologue ST3P are the pheromone mating response factors in fungi generally. And the way this works is that you have two mating cell types, MAT-A and MAT-alpha. MAT-A cells secrete the A factor pheromone, which is a short 10 to 12 amino acid peptide. This will bind selectively to the ST3P receptor that is only expressed on the MAT-alpha cells. MAT-alpha cells will secrete a different pheromone, the alpha factor, which is just a different peptide, which will only bind to the ST2P receptor, which is only expressed on the MAT-A cells. So in both instances, binding of the pheromone to the receptor leads to classic signal transduction, including stimulation of a heterotrimeric G protein, so you have dissociation of the alpha subunit from the beta gamma, and stimulation of a mitogen activated protein kinase cascade, which ultimately leads to modulation of gene expression and cell cycle arrest. Once the cells have arrested, they're then poised to undergo the second portion of this process, which is the actual mating events, where you have projection or smooth formation. So this is where the cells deform and start growing toward each other. Cell fusion, so that's the cytoplasm and nucleus actually fuse, and ultimately a diploid cell formation. So this is an example of classic GPCR signal transduction. You have one ligand stimulating one receptor, leading to stimulation of one pathway. So I've been studying ST2P ever since I left Gobin's lab 17 years ago. And most recently, I've been interested in the idea of whether there's the possibility of biased signaling in ST2P. And what biased signaling is a newer concept in the field of GPCR, where for a single receptor, you can actually have different ligands. And those different ligands will actually stimulate different signal transduction pathways through that same receptor, leading to different biological outcomes. So in terms of the work, I did this all pre-wheat. We obtained evidence of a second lower affinity pheromone binding site on the ST2P receptor that actually seems to stimulate initiation of the second part of the mating pathway. At the same time, we also obtained evidence that arrestin may be involved in mediating this effect, although we haven't yet identified what pathway this might be signaling through. So that's what I was doing before, and just keep that in the back of your mind. I'll come back now to Fusarium graminearum. So about 18 months ago, in 2015, late 2015, this paper came out in Nature by Antonio Di Pietro. He's a Spanish fellow, and he's working on Fusarium oxysporum, which is a little different than graminearum. It's a soil-borne fungus, so the spores remain in the soil, and when they germinate, they grow toward the roots. And what Di Pietro showed is that the roots of this tomato plant actually secrete a peroxidase enzyme, the catalytic activity of which stimulates ST2P, leading to transduction down a different pathway, a cell wall integrity pathway, ultimately eliciting the chemotactic response toward the roots. So I was so excited when this paper came out. Here it is, evidence also of biased signaling for ST2P. But more importantly is not the results itself, but the questions that any good finding yields. You find this, you make a discovery, there's 10 more questions. And so these are the black boxes. So first and foremost in my mind is what is the nature of the ligand that the peroxidase is producing that's going to be stimulating that receptor? And what is the effector that's mediating the signal from the receptor down to the CWI pathway? Does this extend to other fungal species that infect other plants? For example, graminearum. Does the wheat head secrete a peroxidase? Is there a CWI pathway? Chemotaxis toward what? If the spore is already sitting there on the wheat head, what would be the point? What about getting away from plants? A human fungal pathogen, Candida albicans. Humans also do produce peroxidase as an immune response, so it's a possibility. And in terms of Cerevisiae, is the CWI pathway potentially mediating the biased signaling that we're seeing in the mating response? So lots of questions there, probably to see me through to retirement, I hope. So we wanted to investigate Fusarium graminearum, so we set up a chemotactic plate assay. Essentially, we have just a layer of agar with water agar with spores embedded in it. We have a scoring line and two wells, one on each side of the line, with a test compound that we're going to test chemotaxis toward, and then a solvent control. We let it sit for about 12 hours. We count up the number of hyphae that grow toward the test compound compared to the solvent control, and from that we can get a chemotropic index, where a positive value means chemotropism. So the first things that we tested were nutrients. We assumed that we were doing this more as sort of a positive control. Oxysporum showed a strong nutrient response. Graminearum did not, much to our surprise. There was no significant, to these basic carbon and nitrogen sources, there was no significant response. Except methionine, which is interesting, and I'd be happy to talk to anybody about that at dinner. So we were concerned that maybe the system wasn't going to work, and so then we tested the pheromone as another positive control, and this time we did see a strong response to the graminearum pheromone, not the Cerevisiae pheromone, showing that it is a specific response, so we felt we could move forward with more experiments. So of course peroxidase was the main target, so we just bought some from Sigma, horseradish peroxidase, so we could get on with it quickly. And indeed we saw a strong response to the peroxidase. This was eliminated when we incubated it with proteinase K, or if we boiled it, showing that it really is the enzyme. And when we added a peroxidase inhibitor, this also eliminated the activity, showing that it is the actual peroxidase activity. You need active catalysis there to get the effect. So what about wheat? Going back to the wheat issue. So here, as you can see in this picture, we grew some wheat, and we wanted to see whether there was chemotaxis toward the wheat heads. So we tipped the wheat plants over and put the wheat heads into our plate